So I'm going to try and give you a good introduction into the topic. I'm going to tell you about what we're doing, tell you about what we'd like to do in the future. It's a very interesting space to be in. So as an overview of what my team do, we're interested in integrating electronic devices with biology. But I started my career as a biologist, so instead of trying to make the biology fit the electronics, I think of things from a different way. I want to fit the electronics to fit the biology, which I think, in my view, makes a lot more sense. But here's where we're going to start. So I promised there would be a slide about design rules. So those of you who know who George Box was, he said this: all models are wrong, but some are useful. And that's our mantra. And if you keep that in your mind, I think you'll do very well. Interestingly, despite the fact that George was a statistician, he was interested in species differences. He was talking about mice and tigers, and that's something that we think about quite a lot, species differences. And if you think about the models that are used currently for therapeutic development, there's a lot that is left to be desired. So I don't know if any of you saw these headlines. So December 2022, Joe Biden signed a new law which means that you no longer have to do drug trials on animals. So typically you have to do a small animal model, a large animal model before you can get into clinical trials. You don't have to do that anymore. Now, that's not to say that tomorrow there will be new pipelines starting with no animal models. It just means that the potential is out there. And just to give you an idea, for some cosmetics, because of societal pressure, the gold standard for testing those cosmetics is now done for, certainly for some skin applications and some eye applications, is done on in vitro models. So this is a huge call to arms for people who work on in vitro models. And I think it really challenges us. So new law welcomed by animal welfare groups, but change won't happen fast. Why? Because the in vitro models we have now just aren't good enough. Right? They're just not good enough. So another interesting review, if you think about how we evaluate things in vitro, this is how we've done it for a long, long time, 110 years or so. The new hope is organoids, but as I'll show you, organoids are inside out usually. So it does limit their utility in some cases, but very, very interesting and hopeful. Here's a space, well, we're kind of between this space and this space, microphysiological systems, sometimes also called organ on chip, depending on how you think about it. The idea to instrument your model, to integrate sensing, to have circulation that would be, if you're a pharmacologist, you think about ADME, absorption, distribution, metabolism, and excretion. And you can do that in an animal model, but you can't easily do that in vitro. But then there was the DARPA project. I don't know if you know about the US DARPA. They do weird and wonderful projects. And they had a body-on-chip project in 2012. And the new FDA document that the law was eventually based on refers to that DARPA project and all of this sort of pipeline of things that's coming online. So that's sort of the motivation. There have been people thinking about tissue-engineered models for a long time. So if you think about somebody has a dysfunction in their body, you want to repair that dysfunction. So you want to put an implant in. Best case, I mean, the holy grail for this is somebody has a severed spinal cord. You're going to insert somehow a piece of spinal cord that's going to make them walk again. But you think in 3 dimensions, and what you're trying to do is, as closely as possible, mimic the tissue that was there to make it be functional and do what you want it to do. So this knowledge is there because these tissue engineers have been thinking about this for a long time. And so what we're trying to do is apply the tissue engineering models to in vitro. And there's a really nice sort of added advantage, which is if we have a lot more of these in vitro models, then eventually the tissue engineered implants will get better, right? Because we just have a lot more data and a lot more examples. So we think about how to engineer a tissue, what sorts of cells to put in, what sort of a structure we're going to grow them in. Is it biocompatible? Right from the beginning, we have to think about that. And in my particular group, we think about how to measure that. A lot of biologists, myself included, are very happy to do what's called an endpoint assay. What does that mean? You grow something for a couple of days, weeks, months, and then at the end, you look to see what happened. What happened during when you were growing? You spent 6 weeks growing this really complicated organ system, but you weren't measuring what was happening during. And if you added something like microbes of the microbiome, how would you know what they were doing over time? So having some sort of continuous monitoring is really important. So if you think about a tissue, this is what I would call a stratified tissue. So we have a barrier layer, epithelium, which is very good at keeping things out and letting things in that should get in, unless it gets compromised. And then you have connective tissue, which is filled up with proteins and polymers and sugars and so on. And this order of things is important. Now, we've known for quite a long time that if you add different biochemical cocktails, you can get cells to do different things, to grow or differentiate in a particular way. And then roughly 30, 40 years ago, the idea of mechanotransduction, the fact that cells would change their fate depending on the stiffness of the substrate, that became quite clear as well. Electrical cues is something we think about a lot. Well, there was Galvani in the 1780s. He thought about it a lot. He did those crazy experiments with frogs attached to lightning conductors. He was thinking about it, but then it kind of got a bit lost. But we know that, for example, between different sides of a wound, there's an electric potential there. How do the cells know which direction to grow in? Different tissues respond to electrical cues in different ways. You can have fast-growing cells and slow-growing cells, and it could be because of their membrane potential. So neurons have a very different membrane potential than, for example, fast-growing cancer cells do. So what's our technology? What do we make and what do we measure? So just a tiny bit of introduction into conducting polymers. So for me as a biologist, being introduced to these particular types of electronic materials was key. Because what it meant was that my idea of adapting the electronics to the biology was actually possible. So these start out life as a solution. And what that means is you can spray them, you can knit them, you can weave them, you can make them into scaffolds that I'll show you. You can make them into freestanding conformable films, or you could make a standard electrode by putting a thin film on a device if you want to. What can you use them to measure? Well, this particular type of electronic material is very cool because it has what we call mixed conduction. That means it conducts ions and it conducts electrons. If you know, well, even if you don't know anything about biology, I'll tell you, ionic flux is everything to biological systems. If you don't have ionic flux, it's usually a good sign that your biology is dead. So, ions flow between cells, into cells, out of cells, between tissues. And these materials can pick up these ions and convert them to an electronic signal. And vice versa, actually. So, first of all, these devices were used for neural activity sensing. What's an electrical thing in biology that I can measure? Neural activity, that's fine. But we do a lot of what's called impedance sensing. What does that mean? It means that if cells or a part of a cell or a tissue is near or on an electrode, we measure how many ions are entering that electrode. And if we have a barrier tissue like the one I showed you earlier, or some other sort of adherent cell or other properties of tissue, that ionic flow into the device is going to be modulated. Now, this is all very much shown in 2 dimensions for the moment, so it's a bit hard to conceive of how we're going to do that to fit with our tissue engineering principles, but hopefully I'll show you how we do that. So we started off with these conducting polymer solutions, and we use a process called freeze drying. So essentially what we do is we pour a liquid conducting polymer solution where we add various additives depending on the application, and we freeze it. And then we sublime out the ice crystals, and what we're left with is a honeycomb structure which has pores in it. Now it's a little bit like, if you allow me some artistic freedom here. It's a bit like a tissue structure. And cells can grow inside, they can line the pores, and it's quite well known in tissue engineering circles what the optimum should be. But because this is an active material, when the cells grow inside, what they do is they're impeding the ion flow. And as they grow, they do that to a greater extent. As they differentiate, they do that to an even greater extent, which means that we have a continuous measurement over time of what a cell in a population is doing. So impedance monitoring. Now, impedance monitoring of cells has been known since Kiefer, upstate New York, got a Nobel Prize for, I don't know, electron tunneling or something. GE then gave him a lot of money to do whatever he wanted, and what he wanted to do was cell impedance substrate sensing. But that was in 2D on an electrode, and we were making this 3D. So now to the topic of the evening, how are we going to use this to study the microbiome? So very quick, 30-second introduction to the microbiome. I got interested in this for a personal reason, like probably some of the other people in the audience, because I had been ill, and then after that I just didn't feel right. So I have the luxury of being a researcher, and not only was I able to research these conditions, but then I thought I can actually do something about it. So what happens if if something has happened to perturb your microbiome? Well, lots of different things can happen. It really is something that starts at birth with how you were born. For example, cesarean birth could be linked to dysfunctional microbiome. Antibiotics that you may take during early life, pathogens that you may experience. You've probably all heard of C. diff infections, for example. And then a change in your microbiome, where the best summary I can give you is that the more diverse your microbiome is, the better. And so a change in that diversity can be very problematic. So when you have an altered microbiota, there are effects on the gut. So you have this microcosm. You have all of these trillions of bacteria and viruses and even fungi living in very usually good harmony with your cells. However, when things go wrong, then the cells, the host cells, start to, it's almost as if these bacteria, which were hitherto friends, then start to behave a bit more like what a pathogen would look like. And that can lead to things like inflammation, and then ultimately you can have other diseases resulting from that. But it's not just at a local level at the gut, it's also affecting the brain. And I'm going to show you in a minute how that can happen. But changes in microbiome have been linked to all kinds of things, including stress, anxiety, depression, autism spectrum disorder. So it's possible to study, and in fact, a lot of people do study the microbiome in an animal model. So you can get these germ-free mice that have been raised without any exposure to pathogens. And even, I think, the mothers aren't exposed to pathogens either. These are very, very strange mice. They are often obese. They have very strange behaviors and a very altered, I would say, biochemistry. There have been some clinical trials too, and in fact now for a probiotic to be certified as such, it has to have gone through a clinical trial. So to make those claims that they like to make on the bottle, it has to be certified. So we really felt that there was a motivation here to do some of this work in vitro. So what could we study in vitro? So the initial plan was to have a model of the gut, a model of the brain, to connect them, and then look at a couple of things. So how can the brain be affected by the gut and the microbiome? So one way could be the bacteria are metabolizing things that are eaten by the host, and then a metabolite maybe enters the epithelial layer, which is the barrier layer. And then it gets into the bloodstream, might get through the bloodstream, go across the blood-brain barrier, which is this highly effective barrier that you have, luckily, at the vascular level in the brain. Second way could be through things like hormones, neurotransmitters. Another way is the enteric nervous system. Who knew you had a brain in your gut? You do. You have a lot of neurons in your gut, and the enteric nervous system connects via the vagus nerve to your brain. So that's another way for the signaling. This, as you'll see when I come to the end of the talk, this was the one thing which even I remember the reviewer saying, how's she going to do that? It's very difficult in vitro. So lots of different ways for the gut and the brain to connect. The immune system is also very important. So if we have our electronic devices too, we can measure very interesting properties. So one thing is we could measure hormones and neurotransmitters. We can, of course, using techniques that have been out there for quite a long time, measure neuronal activity. But the thing I'll focus on most is this impedance property. So if we have a good, intact, healthy barrier, if you have a good, intact gut epithelium, then you measure a really nice property called the transepithelial resistance. So the cells are resistant to ion flow. You can do that at the level of the gut, but actually also there's this nice barrier at the level of the brain called the blood-brain barrier, and you can also measure using impedance the permeability or lack thereof of this brain barrier. So really impedance turns out to be a good tool. So we took that scaffold-based electronic device, and in the first generation, this is, when you show people pictures of your baby and then you realize that it's a little bit ugly and clunky, but this was the first generation device. What we decided to do was to make the scaffold with a tube through the center. We were trying to mimic the overall structure of the gut. So you see this is an SEM, a scanning electron microscope image of the scaffold material. And for any of you who are keen electronic engineers, we did the proper device characteristics. You see, if you're familiar with these types of data, this is complex impedance versus frequency. What's very important to note, which gives you a very good signal to noise ratio, is that these have low impedance. It's because they've got a really high volume, so you have really nice dynamic range, if you like, for sensing. Now, we made this device inside a tube because, as a biologist, we knew we needed to feed the cells, and then we put it into a well plate. So biologists like to work in well plates. So it's a very standard format that everybody knows. It fits on all the devices, the microscope stages, etc. Then we used a strategy to build up our gut. So we're using cells. We're using a robust approach to engineer the gut from the bottom up. So we use a type of a cell called a fibroblast. This is like an adhesive secretion factory. These cells entered into the scaffold. They produce tons of this extracellular matrix, which is lots of proteins and sugars that's found in the connective tissue. And then that acts like a landing spot for the epithelial cells. Now, we use two types of epithelial cell. One type produces mucus, and this will become important later. And then the other type is just a well-known cell type of the gut epithelium. So it's important. We start with the TIFs, we leave it 4 days, and then we add the epithelial cells. These are images it's like an in-depth look into the cells. What was amazing to us was that the cells auto-organize. You give them the right cues, and they go where they should go, and they do what's called polarization. So they grow tall, their nucleus goes down to the bottom, they project these microvilli structures at the top, they push the mucus out to the top. So that means you have a lumen lined with mucus, which, like I said, I'm going to tell you why that's important later. You can see the mucus pointing on the top here. So this was really nice proof for us that we could use this tissue engineering strategy. This is, something that could be developed in the lab and used then for testing. But it wasn't even remotely high throughput. It was tricky to build. We were getting 3 per plate. It was tough going. So we had to really think how to make this useful, because if you want to do biomedical research, you need lots of replicates. You have to have a lot of data. I missed the punchline from the previous, which was, this was the optical imaging, and really importantly, we had continuous electrical monitoring, so it wasn't that endpoint assay that I told you about. And this was, earlier I told you that the scaffold looks a little bit like what you might expect a tissue, connective tissue to look like. I think the best thing about this image is that the scaffold doesn't look so invasive, because one of the criticisms we get is you're building synthetic tissue. But the scaffold is here, it adds functionality. Here we have our fibroblast sort of in a stromal layer, and then we have the epithelium on top. Okay, but now I'm going to get to the getting more throughput into our devices. So what we thought we could try, we really want to keep that stratification, because for anybody doing any kind of a test, you want to be able to add something on top and then look at the transport through. Organoids don't allow you to do that. Organoids, you have to inject things into the center because they're inside out. So that stratification was really important. And there was an existing device called a Transwell, well known to people who work on any kind of barrier tissue like blood-brain barrier, skin, kidney, or gut. And it's a hanging insert made of usually some sort of a plastic like nylon with pores in it. And if you grow cells on top, they can feel cues from underneath and from the top, and so they differentiate in a more in vivo-like way. What we did was we replaced the nylon with a slice of our scaffold. So this would allow us to build up a pretty 3D structure, give us stratification, but it allows us good access to either side. And the advantage over the previous Transwell device is that it's a functional device. You have the built-in assay, if you like. So again, we do characterization on the device. We have nice low impedance. We worked out what thickness was best to make sure we could get a good tissue. This is some of the steps we went through fabricating them. We're now in the process of trying to figure out how to prototype this, because this is all manual using kind of watchmaker technology. It's not the, it's an academic lab after all. Can we build up tissues? Yes. In this case, we built up a gut tissue and actually something a bit like a vasculature. So again, our strategy is fill the bulk of the scaffold with the fibroblasts. That gives you a nice bed to add your either epithelial cells, here, or endothelial cells. The images just show you nice evidence that the right cells are there in the right place by staining for markers that we know should be there if the cells are differentiated. So these are, again, the two types of epithelial cells. This one produces the mucus, which you can nicely see in various different patches. And then the HUVECs are, it's a model for endothelium. Here's the electrical monitoring, and we're starting to build up signatures for different types of cells. So biologists like to image things, and then they see the outline of a cell, and then they're reassured that that cell has a morphology that they would expect. So when we're trying to convince people of our electrical monitoring, we have to do a lot of work to validate. We have to say, look, all of your regular markers are here, but look at this additional biomarker we're giving you. We can measure electrically what the cells are doing over time. And so we're measuring every day, sometimes more than that. The fibroblasts gave us a very interesting signal. I can tell more of you about that. We're doing more work in the lab to figure that out. But we don't see a characteristic impedance change in the frequency we'd expect for cells like the barrier cells, where we see it's like it goes from being a hockey stick to a pregnant hockey stick. So here in this mid-range of frequencies, and this is in literature, we expect a change in that range. The HUVECs, it's not well known in literature because they're super hard to measure, because usually the sensitivity is not near where you want it to be. But don't forget that it's as if we have electrodes all over the place, because we have a three-dimensional structure of electrodes. So anywhere a cell sits, we're essentially improving the sensitivity by approaching the electrodes closer to the cells. We can do some functional assays to show that if we add a compound, this particular one chelates calcium, what happens is the cells open up. And that opening should give us a characteristic change, which it does. This is a reversible assay, so we can do it continuously. So we add the EGTA, and we see a decrease. And then we essentially replace calcium by changing the media, and we see an increase again. So it's a functional assay that really is working dynamically. So a sort of a proof of concept experiment was to take these transmembrane electronic transmembrane devices and use them for a particular purpose. So I've shown you already the characterization. And again, we have these nice polarized cells. We have a continuous layer, which is important. And we use them to study the microbiome. So we've been working for a number of years with a group in UCC in Cork. So it's the APC Microbiome Institute. John Cryan and Colin Hill are internationally known experts on the microbiome. And yes, I had to cold call John Cryan and ask him if we could collaborate. And they've really been wonderful collaborators. And the first thing we decided to do was to look at the difference between what's called a postbiotic and a probiotic. In other words, does it make any difference if the bacteria are alive or dead? So if you look again at your probiotic, you might want to know, are they live, so like a Yakult or an Actimel, or is it dead? And it does seem to make a difference. So what's really key here is that most cell biologists won't let a bacterium within a mile of their cell culture because it contaminates them and it kills the cells. But our bodies are very used to having bacteria in contact with our cells. How do they do it? Well, it's because probably the bacteria aren't actually contacting the cells. They're sitting in that mucus layer above the cells, and they don't penetrate into the cell layer, and they shouldn't, only if you have really bad, I don't know what it's called, when you have really open, permeable epithelial layer, the bacteria can enter into the bloodstream, and that's never good. But so our bacteria, we tested it, they don't go through, they sit relatively happily for a limited period of time. I will say that, because eventually the bacteria could overgrow, because we're growing in full oxygen conditions here. Anyway, so we used a cohort of microbes called SAHUMI, Simplified Human Microbiome, and these have been very well characterized and sequenced and so on. 4 are known to be pro-inflammatory, 4 are anti-inflammatory. They're well known and characterized because it seems that what you put in is what you get out. So in a lot of microbiome studies, things change as you transit through the gastrointestinal tract. It's as if you're expecting, the pH is different pretty much everywhere, the oxygen conditions are different, the nature of the tissue is different. So it's quite unusual to have what you get in being the same as what you get out. But anyway, this particular cohort maintains certain ratios. And some are known to be pro-inflammatory and some are anti-inflammatory. So this is postbiotic. And why is it postbiotic? Because it's very hard for us to maintain those ratios growing in the lab, because we can't grow in the oxygen conditions that would maintain that. We're growing and cultivating cells at the moment in relatively high oxygen. So we add these bacteria, either pro-inflammatory, anti, or the whole thing, and what do we see? We see a decrease in the integrity of the cells. These are actually not doing any good, they're doing harm to the cells. But don't forget, these are dead bacteria. The bacteria in your gut are hopefully alive. So how do we check that? So the one bacterium that we know can grow very well is the commensal E. coli. It's able to grow in oxygen conditions that the majority of labs have available to them. And this is a serious failing of microbiome research, by the way, is that if you're growing things in oxygen conditions that we typically do our cell culture in, it's just not right. Normally what happens is that the tissues perceive oxygen from the blood below, but the apical surface wouldn't have, a high sugar, high oxygen, high everything media on top. Anyway, the E. coli, the live E. coli, so this is just a lab strain. This one here is a strain from the SAHUMI cohort. When they were alive, we see an initial decrease, but then they recover. Something about the bacteria being alive really makes a difference. So this is a key finding. We tried F. prausnitzii. It's a strict anaerobe. We tried growing it anoxically and adding it, but we think what happens is the bacteria died. So it was as if there were no bacteria at all. So we're not even sure we added bacteria in this case. So both E. coli of this strain and that strain, we see a decrease and then a recovery over time. Okay, so how am I doing for time? Good. So up until now, our model is still fairly reductionist. So we want to get to this stage, but in the gut, there are a lot more cell types, and there's that immune connection, there's that neural connection. So we thought it would be good to try and increase the complexity of our model. So we have a model that works, but there's still bits missing. And so people say, well, what about the immune cells? What about the neurons? So sometimes when we want to develop the model, we go to the transwell. So this is the transwell that I spoke to you about earlier. So it's this porous nylon filter. But to give us the 3D nature, we replaced our scaffold with a gel made from collagen. So you can see collagen, it's a cosmetic product that people drink for an odd reason. But we used it and we put immune cells in that gel. So this is again a cell line, so this is a robust model. And we can either keep them as monocytes, which are non-activated, or we can make them activated macrophages. And then we put our epithelial layer on top, and we measure, this time with a commercial device, because we don't have our scaffold, we measure this transepithelial resistance, and we see that the sort of more complicated, more representative model gives us this enhanced barrier integrity. It's a sort of if this is your marker for how good your model is, then the one with the macrophages is the best model. We then added the bacteria, and like what we saw with our scaffold devices, with the macrophages, we see an initial decrease, but then a recovery. These are live E. coli, by the way. They're green, which means that we can see them. You see how they're sitting on top here almost? Sometimes there's a separation. They really don't enter into the cells. This red is not mucus. This red is the top of the cell, and you can see that the E. coli are sitting, so maybe the mucus layer is in between. So again, recovery if we have this activated immune cell. And then because we hadn't done a proof of principle on these transwell models, we worked with Cinzia Cantacesi, who's at the vet school, and she's interested in worms. It's frowned upon to give people worms, although it's being done more and more. But the problem is if you don't have a human host, you can't really grow them very well. So there's a real need for in vitro models. And they literally collect worm juice. So they grow the worms, they collect the secretions, and those secretions are thought to be beneficial for health. So this is why people are interested in worms. We're too clean. When we got rid of all our parasites, we lost something. But it's something about the interaction between the worms and the bacteria. It's not enough to have just worm juice or just bacteria, you have to have both. And to cut a long story short, when we use the model I just showed you, we add E. coli and we see recovery. But if we add the E. coli plus the worm juice, we see a faster recovery. So there is an enhanced effect that we see. And we did, in this case, cytokine analysis. It was very cool. Anthea, who did the images, was able to image, get a stack of images. And what she could see was evidence that the macrophages are able to move through this collagen gel. They have to be mobile cells, they're not fixed. And they were sampling, almost as if, there's something going on, so they're moving up towards the epithelial layer. At least that's our hypothesis. So that's including immune cells. We're now including neurons. Well, neuron-like cells. So these are cells, it's again a cell line. It's all been cell lines up to now, so readily available tools. There are critics who say cell lines aren't good, but they're good when you're building an engineering model with engineers, believe me. So these cells, we put them in the gels, and already, even without the regular differentiation protocol, they were getting happy and activated and extending these spines or dendrites in 3 dimensions. A lot of people grow neurons flat, so not actually in 3D. Although I'm on sabbatical, so I'm doing a lot of reading, and I just read that Harrison, who started cell culture basically about 1908, he grew his neurons on fibrin clots, so he actually had a 3D culture. We just forgot what he did in between 1908 and about 1980. So we're including neurons. We've shown that those are active. And now we're working with Matthias Zillbauer. This was an initial collaboration with the Milner Institute. And Matthias Zillbauer at Addenbrooke's and the Stem Cell Institute has organoids from a variety of different patients, some with IBD, some without. And what they do is they expand the organoids. So organoids are fantastic because they have loads and loads of cell types that you naturally have in your gut epithelium. So they expand those, and then what we do is we dissociate them. So we take the organoids and we open them up. And then they lay down on our nice bed of extracellular matrix, and we get this nice polarized layer. So what we're doing now is including those cell types and trying to do some of the studies I showed you before, but now on organoids. And then we'd be able to look at, say, IBD or non-IBD in pediatric and adult. Okay, so that has been the gut. And you'll laugh if I tell you that was the easy bit. The brain, yeah, we nearly gave up the brain a lot of times. And when I say brain, what I really mean is neurovascular unit. So we're looking at this interaction between brain capillary endothelial cells, which feed individual neurons, and then you have some other cells like astrocytes. So this is very, very complex, 3-dimensional architecture. But what people do, us included, is we collapse it down to a stratified version on a transwell. We know it's important to have the cells touching and close to each other, but this is really, really hard. So to get the cell types right, at least, we developed an in vitro model on the transwell. So this is our neuron-like cell, this is our astrocyte-like cell, and this is our endothelial cell, so the blood vessel cell. We developed a protocol, and then we measured using a couple of different parameters. The resistance is never very high. There are reports in vivo that it can be about 1,000 ohm centimeter squared. I have my doubts, but anyway, in vivo, most reports never go above about 50 to 100, except a couple of reports which, again, stand alone, so need to be validated. You can also check permeability. What is the rate of markers going across? And that's a standard biochemical assay. So it seemed like our triculture model was working. But then we wanted to transition that to our scaffold devices. So again, brain more complicated. We need different types of scaffolds. But remember, I told you we start with a liquid conducting polymer solution, so we can add stuff. We're like mixologists, cocktail shakers. We can add in molecules that we know are important for neurons to adhere. So we added in hyaluronic acid, collagen, laminin. And we made scaffolds, which presumably expose these proteins for binding for the neurons or other cells to bind to. And then we adapted the differentiation protocol and characterized. Now, it's important for our devices that the impedance doesn't change too much when we add these polymers. So does it deteriorate our device? Not really. Interestingly, the devices, when you add the biopolymers, are a lot softer. This is important for brain tissue because it's got the consistency of jelly. So we're still a bit high for brain tissue. So at least we're approaching something, I think about 2 to 4 kilopascal is about right for the brain. So we characterized the neuron-like cells after the differentiation. We looked at properties like their ability to sprout and different markers. But I think possibly the most interesting data is we worked with an electrophysiologist at the University of Reading, that's Francesco Tamagnini. And if you compare the basic scaffolds versus our, cosmopolitan dirty martini here, then you see we have a much more mature neuronal phenotype. Essentially, the cells are responding to the biocues and maybe going further down that differentiation pathway. So it really matters what you put into the mix. Okay, so we have now made scaffold slices and are putting the various different cell types into the scaffold slices. Good news is they grow, so these are the capillary endothelial cells. Here they're lining the pores, which is not necessarily what we want. This is more of a biocompatibility test. What we want is, again, probably a stratified version. But I was just talking to Chiara, who's working on this project yesterday. Maybe we should allow the cells auto-organize and see, what happens. The problem is you want to be able to access the capillary to be able to add compounds and see what happens to them. But we're still working on that bit. So this was the capillary endothelial cells, and this is then a mixture of astrocytes and neurons. And it really seems like the astrocytes do what they're supposed to do. They support the neuronal growth. Here's yet another signature. You should be familiar with these by now. So the classic baseline, hockey stick, and then this sort of shift at the mid-frequency range when we add the endothelial cells. And this is hot off the press, about 2 days old data. When we do the triculture, we're still trying to figure out where the different cells are. But it looks like we have evidence that we have a probably very, very wavy layer of capillary endothelial cells, but definitely astrocytes and neurons all projecting in 3 dimensions. And again, very new data just showing that when we have the triculture, we do measure this significant change in the resistance over time. Probably it peaks and then comes down again versus a control where we have no cells. So what's next? So I've shown you human in vitro models. We also do, by the way, have a rat in vitro model. This was an idea. I went to a conference on alternatives to animal technologies, and it was Roche Predict. So it was for post-market surveillance. After you have a drug and you've done all the clinical trials, and then there are these weird side effects, how do you explain them? Well, one of the ways they were suggesting was you have in vitro models. It's more like comparing like with like. So we're developing rat in vitro, but we also are looking at ex vivo. So we're trying to compare some of our in vitro models to what we could get with tissue from rodents, because that's more accessible. And we often use mice or rats that have been used for other experiments first. And then we can compare it to in vivo. Now, I just had a message from a collaborator in London that we'll have access to human ex vivo tissue in a couple of weeks. This is people who have undergone bariatric surgery. There can sometimes be additional tissue which otherwise would be disposed of. So what I'm going to show you now is how we're going to measure the ex vivo tissue. So same materials, but the device format has to be now different. And we had done some work previously where you can actually buy, this is, it's a cartoon of tracheal tissue. Tracheal tissue is interesting because it's grown at what's called an air-liquid interface. So in other words, you have air above, you're supposed to have air, and then liquid below. And the problem is that if you try and measure electrically from the top, you don't have liquid. So you have no ionic conduction path or electronic conduction path. So the thought was, if you had a conformal device, it could sit on the mucus of the tissue, and then because you have an ionic pathway, you'd be able to measure something. And we had done this to look at the effect of electronic vaping, so by puffing in this electronic cigarette vapor on top of the tissue. It does really bad things to the barrier permeability. We were a bit surprised by that. Anyway, that was another story. But so we've adapted that device design. We've made it a bit better, of course. And now we don't need opposing electrodes. We can just build everything into this conformal, planar electrode that will sit on top of tissue. We characterize the device, and then we added it to this is in vitro. This is so just CACO2 cells grown on a transwell. And we again do the EGTA assay. So we have a barrier, then we add the eGTA and it goes down. So this is the measurement from our device, and then this is a commercial device where you have to add liquid on top, and then you measure the resistance, and we see that we got pretty comparable signals. So we're very happy about that. Sophie, who's the student working on that, had a look. So we have a collaborator in pharmacology, and she had a look at how that's done now. You know, I think her goal was to find all of the really archaic instruments and say, we can do that smaller and better, and you know, just thinking about what things she could measure. So we can measure impedance, and now she's been focused on taking whole gastrointestinal tracts and measuring contractility. So contractility is very important for gut function and impedance and enteric nervous system. So this is the thing we couldn't do in vitro. So what I've focused on most of the time talking about the gastrointestinal tract is this mucosal layer, and sometimes a little bit of the underlying layers. But the ENS connects to this mucosal layer, and then, like I said, maybe via vagus nerve or other ways, it then signals the brain. And we still have not thought of a way to do that in vitro. So we've been thinking of a series of devices that could do that, at least ex vivo, and a bit in vivo now too. So this is again with the team in Cork, and also George Maliaris's group. So Amparo is from George's group, and then Alex is a postdoc who works between our groups. And so he's been focused on accessing the submucosal plexus, which is a layer of the enteric nervous system that we think we can access. Now, it's all embedded in various layers of muscle, so the first experiments relied on stripping off some of that muscle layer and thinking how to do it. I'll just skip this one. So these are the devices, the initial devices anyway, that Alex built. So instead of a conformal electrode, he wanted a sort of a pronged, imagine like a paintbrush approach. So it's going to go into the tissue and try and hit and get recordings of neurons. The ENS has fewer neurons than you would find in the CNS, and they're not as well mapped, so you don't really know where you're going. And we're hoping we're at the submucosal plexus. So he developed these devices, and just let me run you through this. So what this is an entire gastrointestinal tract sitting in what's called an organ bath. So this allows you to keep the gastrointestinal tract alive. So you're perfusing it with what it needs to stay alive, at least for, and I think they can stay alive for maybe 24 or 48 hours maximum. And then he stripped off the muscularis layer, and so we should be sitting directly on the submucosal plexus. So this is his device, and it should be sitting on that layer. And then recording. So this was the first attempt. This was actually done in Cork. Now we can do it here. The Cork people had all the expertise in how to, excise the tissue and access it and so on. So the easiest thing, or the most hit with a blunt instrument test we could think of, was distension. We thought there has to be a neural response if we distend the gut. So just put a bolus, clamp one end and put a bolus of fluid so it just expands. And it looked like the time signature seems to correspond with that. So we think we saw this evidence. But this is still relatively preliminary data, just showing you that we're trying to access, using these new tools, things like the ENS, which has been surprisingly underexplored. So at the moment, I told you we have to think about oxygen. We have to think about growing anaerobes. We also have to think about growing the tissue with oxygenated media below, but no oxygen present. Most cell culture it's not mimicking blood very well, and in fact the cocktails for the different cell types are all very, very different. So when you have co-cultures of cells, you have to optimize it for each cell type that you're putting in. We're also trying to access metabolomics, because those studies I showed you earlier, we could do much better if we had readouts of what the bacteria and the cells are co-metabolizing together. Alex and Amparo are now doing in vivo GI measurements, ENS measurements. And one thing they're doing is they're clamping the vagal nerve and then looking at how that affects ENS. We're trying to think of various different stimulants that will affect neuronal signaling so we can know for sure that we're measuring a response. So I'll finish there. I showed you a lot of work from a lot of great people. Costadinos unfortunately had to leave because he has to go to an enterprise tech event. But he and I and another student, Doug, we're in the process of starting a company for the electronic transmembrane device, just because we really feel there's a need for these in vitro devices. Lots of funding from very kind sponsors, and thank you to you all for listening.